Yay.
Okay. Some of us have made it.
I'm just gonna email it to Shannon so she can get on too.
Thanks, Christopher, for doing that.
Yeah, no problem.
For real. I hate Hangouts on Air. I never could get it the whole semester. Like,
I
it
suppose
sucked.
you can all thank me after we know it worked.
I'm sure it will, though. I'm not even worried. Okay, I just sent it back. Like, I just emailed it to Sarah just in case she didn't see the chat, but I'm trying to email it to Shannon too. Okay.
Oh, I think this is the one you can add stuff like sunglasses so it floats on your face.
Oh gosh. So useful. Oh no. I'm still trying to
Okay.
get Shannon on, but so long. I don't know.
Yeah, we lost Jennifer. I just sent her the link.
No, Jennifer's on.
I'm
Oh,
back.
Jennifer's on. Oh, okay.
Hmm.
Live. So I think we're recording now.
Yeah,
Yeah.
we were.
We lost someone though.
Well, we're waiting for Shannon, but I just sent her the link. I just emailed it to her, but she can't find it. Does somebody else want to email her that?
Yeah.
Hmm.
Let's see. I think I have her email. Yeah, I'll send it to her.
Okay.
So was it the introduction group problems that we're supposed to answer together?
Yeah, I'm pretty sure.
And I think we probably have a lot of leeway this time. I think we're supposed to do it before, and then in this group we just discuss anything that we had a hard time with, or work through them together.
Oh, okay.
Let's see here. It says please complete the worksheet before your scheduled meeting, but then it also says as a group you will work through each of the problems.
Okay. Yeah, I started working.
I have them finished.
I was still working on the very last one.
Yeah, I still have number 5 to do. I was in the middle of working on it, but I don't understand 3 and 4.
Yeah, I haven't done these, but I have them open and I'm like looking at them and we'll see how this goes.
Oh, Shannon's here.
Cool.
Shannon.
Party. Squad. Okay. Okay, should we just get started then?
Yeah.
Yeah.
Okay.
All right. So, were there any terms that anybody had a hard time with?
I don't know,
Speak
I'm
now.
just looking at this.
Not really.
I did.
Okay.
The difference between just a simple chromosome set and homologous chromosomes.
Okay. So far as I understand, the chromosome set is the entire box, whereas a homologous chromosome is just the actual little chromosome that has another homolog with it that matches with it. So it's just the individual chromosome within that. The chromosome set is all 23 pairs.
Oh,
Oh.
all
So then
22.
is the homologous a homologous chromosome is just one or is it two?
So the wording's really weird because sometimes they mean both and sometimes it just means one. But a homolog just means that it's the one thing that's identical to another thing. Like how you could have chromosome 14 from dad, and there's a homologous chromosome from mom as well.
Okay.
Okay, I found the one we're looking at. There's another document that was kind of confusing. Okay.
Okay,
So
and then
you said the.
what's a
what?
sister chromatid too?
So a sister chromatid is when a chromosome is copied, so like just one chromosome, and then sister chromatid that has an extra chromosome on it. It's been copied, and that's when it looks like an X.
Oh, okay, okay.
So those are identical.
Okay.
So do you know the rest of the terms?
Yeah.
I think I'm
And
good.
so someone said the group set was just the 24 chromosomes then?
Yeah, humans have 23 pairs or 46 individual chromosomes,
Okay.
and that's the whole chromosome set.
Okay, should we go over 2 and 3? Because those were the ones we turned in last night.
Yeah, just the drawings.
Yeah.
Mhm.
I don't know how you guys want to do that. Just hold the picture up to the camera.
Skull 1, 2, 3, go.
I think there's pictures for that in the PowerPoint. That's all pretty much. It shows you exactly what you need to draw.
Yeah. Oh,
Yeah.
does anyone know when like she gives the grade and it says like a 100 or something and then it has F on it?
Mine says that
Like,
too.
okay, so is that supposed to be like we start as an F or
I don't know. I thought it was funny. I took a picture of it. It's like 100%
yeah.
enough.
I just wondered if everyone else was like that. So number 4 is the one that I had difficulties on. I
Yeah.
took this class last semester and I didn't understand this one either.
Okay, I have answers, but I don't know if they're right. So if anybody else has it done, we can like compare. Does anyone have number 4 done?
I tried it, but I didn't know what I was doing.
Yeah, I did.
think. Oh,
I did
sorry.
number 4. Sorry.
Okay, I think that we need to get some help from the teacher on this one. I can read what I have, and if you want to contradict me, you can. But if you want to ask me why, I don't think I can give you a good reason.
Okay.
Okay.
Okay. So prior to DNA replication, 48 chromosomes, because nothing's been copied, 24 homologous chromosomes, and 0 sister. Okay. Beginning of metaphase 1, everything's been copied. So 96 chromosomes, 48 homologous chromosomes, 24 sister chromatids. End of telophase 1, you have 48 chromosomes, 0 homologous chromosomes, 24 sister chromatids, because they're like split apart, so they're not together for the homologous. Okay, end of telophase 2, 24 chromosomes, 0 homologous, and 0 sister chromatids. After fertilization, 48 chromosomes, 24 homologous chromosomes, 0 sister chromatids. So same as we started with.
So how do you figure those out?
I get the first one and the last one, but the 3 in the middle.
I can't explain it well. I just kind of tried to look at my drawings that I did and come up with the best solution. But I think I need help on this too. I don't feel super confident about what I wrote in those ministries.
Hmm.
Does anyone know if there's like an exam or anything like that, or is it just the quizzes?
I don't know.
That's a good question.
I thought I saw somewhere in there about exams and being closed book exam.
Oh yeah, under lesson 3 there's an exam 1.
It's probably safe to say that number 4 we're probably gonna send to teach and figure out how we do it.
Mm-hmm.
Because I think I just kind of bumbled my way in there. I understood like the last 2, of course, but I think I got my math all wrong on some of them.
Yeah, I don't know how to do that one at all, so I wouldn't be able to teach anything.
Well, let's just leave it where it is. We'll tell the teacher we need some help. Number 5 is just a monohybrid cross, so you can do a Punnett square for that.
Did everybody get 3 tall, 1 dwarf, and then 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive?
Yes, I think.
I didn't get that.
Um,
I got. Hang on.
Oh, sorry, I did double heterozygous, so you're right. Whatever you're about to say.
Okay, so I got the phenotype is the genotype is 2 heterozygous or half heterozygous, 2 homozygous recessive. Phenotype is 50% tall pea plants, 50% dwarf pea plants.
Yep, that's right.
That's what I got.
Okay, but so it says in a cross between a heterozygous tall pea plant and a dwarf plant. So then you could assume that the dwarf one is homozygous.
It has to be homozygous recessive, otherwise it can't be dwarf.
Okay.
yeah.
I was confused about that.
Yeah, so for the phenotype, would it just be the same thing but with tall and dwarf?
Yeah, so phenotype is what it looks like.
Yeah.
So all the
Okay.
And in this case, it doesn't matter as much, but you just have to be careful because those numbers aren't always going to be the same thing.
Yeah.
But the phenotypes are 50-50, right?
Yeah.
Okay. Okay, that's what I got too.
And for certain number 6 is kind of the same thing, but
Just like more complicated, like a bigger punnet.
yeah.
yes,
Yeah.
I hybrid cross. Does everyone know how to draw that? It shows in the video. I did it wrong before I watched the video.
Yeah, I started it out wrong and then I watched the video and you have to cross them first. So
Yeah.
the offspring can only get one S and one Y type of thing. So you have to make those crosses before you can make the squares.
Yeah, exactly. So I got 3 8s are smooth and yellow. 1 8 is smooth and green. 3 8s are constricted and yellow. 1 8 is constricted and green.
Yep.
Mm.
Yeah, that's what I got.
Do we all understand it?
I guess I missed something.
Those are but are those the phenotypes though?
Yeah.
Okay, so what's the genotype ratio? Do we have to do that too?
No.
No.
Oh, we
Yes.
don't. Okay, that makes sense then.
Yeah.
Okay, I guess moving on. Number 7 was probably the most difficult to number 4. You have I don't even know the name for it because there's 4 letters. So that one you have to do the fork method. I got as my genotype 0.03125.
So how did you do yours? Because I got 1.56.
Yeah, it's 1 out of 64, which is.
Yeah, I got
ends up
1
being-
out of 4 as well.
Okay, I guess I did it wrong. So what were your numbers that you multiplied?
Do it with. I don't know if it'll show up on the screen long enough, or if it'll even show on mine. No, it's not going to focus. Anyway, you make 4 columns, and then you just, and you can tell me to stop talking if at any point you already know what I'm talking about. But you just look at the result of the cross. So in this instance, we're having big A times little a, and then you look at our result, and what we need is a heterozygous. We need a big A, and the odds of that happening between big A times little a, that'll happen half of the time. And then you go entirely new thing, do that same thing for our cross with the B's. And so in our case, we have heterozygous times heterozygous, and that will always also result in 1/2 of those offspring will also be heterozygous, like is in our what we want at the end genotype. And then you do same thing for the C's, but in this case, because our result is homozygous recessive, it's only 1/4 of the time. And then with the D's, since it's homozygous dominant, it's also going to be 1/4 of the time. And then you look at it across, and then you have 1/2 times 1/2 times 1/4 times 1/4. And if you multiply that all out, it gives you the percentage of offspring that'll have that. Or if you want it in the clean fraction, then you just multiply the denominators and then leave it as 1 over whatever that ends up being, and and that just gives you that proportion for the genotype.
Okay, so I had all that except my D percentage. Instead of 1/2, I had, no, you said 1/4. I had 1/2 because it looked like when I did, oh, it's because we want big, okay, I thought we wanted heterozygous. We want homozygous dominant. Okay,
Yeah.
so it's 1/2, 1/2, 1/4, 1/4. Thank you.
So you were able to get the final result from that without doing all of that that method that they showed on the little video.
Yeah,
I
if you
tried
were
doing
to,
that and it was like very long, and I wasn't sure if I was actually doing it right.
to do that, all that would do is to show if they wanted to know the genotypes for everything.
Mm-hmm.
Whereas with this question, since all they ask is just one specific genotype, you can just do your multiplication for one specific thing. Whereas if the question had been, what are the genotypic ratios, then we'd probably be doing this problem for a long time.
Yeah.
I
Okay.
hope we never have to do that.
And that was long. All right, I kind of see what you guys did.
Yeah, if you just make 4 little pundit squares, like you take any of the letters, you just make the pundit squares for each of the letters. So the A's, the B's, the C's, the D's, and so on. That's how I did it, and I got the same answer. And it kind of like breaks it down.
Okay.
So you don't end up with like one giant Punnett square.
Yeah, exactly.
Or a whole list of fractions.
If you ever want to check your stuff online, there's online Punnett squares too.
Oh, okay. Cool. That wasn't too bad, you guys. I feel like, I don't know. I know it's gonna get harder, but
So,
it's so
yeah.
fun.
So what's that document that is under the? Let's see if I can find it. It's called Introduction Lesson Applying Genetic Problems, and it's 1 through 5,
Where's
and then
that?
you have to It's
Oh.
under.
Ponder and prove.
Yeah.
Oh.
So are we supposed to do this as a group? Alright, that's what I was confused about.
I think it's just like individual work, because it seems like we have to scan some of this and turn it in. That's how I thought it would be.
Okay. Because I wasn't sure.
Oh, I think that is individual too, because it says Introduction Lesson Applying Genetics Problems Worksheet, and then it has a work submission folder, and then it has a key review, and then it has the quiz.
Okay.
Yeah, so I think that's all like one chunk that we're supposed to do by ourselves, I think.
Okay, yeah, so it says submit number 3, part A.
Yeah. I think the only
Where
thing
is it?
Say that again.
Where is this?
it's under the Ponder Improve under Course Introduction. So go to Course Introduction, then go to Ponder Improve, and then it's like those 4 things.
In previous semesters that I've had online classes, Ponder Improve was always individual work. But
Yeah. Yeah, I'm pretty sure teach one another is the only thing that we do as a group, like,
Group.
and then everything else is individual.
So then do we do the introduction group problems key review together? Under teach one another?
What?
Yeah. Yeah, we're supposed to answer those together. And like we each individually sent in one of our own.
For the group problems, though, not the
Yeah.
applying genetics. Okay.
Yeah, group problems.
Okay.
Is that under Teach One Another?
Yeah, that's the one we just did.
Oh, okay.
Yeah.
And so then whoever did the air thing will copy the link into that then?
Yeah,
Yeah, in the discussion board.
if you guys are nice to
Okay.
me the rest of the video.
Yeah, so it says that we each do it individually, like we have to do it on our end, but we do it as a group.
Okay.
So we all have to click on the link and answer the little, whatever it says, it's a quiz, but
Yeah.
The key review thing with the question mark next to it, you mean?
Yeah.
Yep.
Yeah.
Oh, okay.
It's the survey.
I think it's a survey about how our
Oh, yeah.
and
You are getting credit for completing this survey.
it says the instructor it will help the instructor understand problems. So he's on it right now.
It says, I answered number 1 correctly, I answered number 2 correctly, yada yada yada.
Oh, so we're starting it now.
Yeah.
Well, we're gonna do it later.
I
Is
said
anyone
yes
in
for everything before.
Rexford?
Yeah, I'm in Rexford.
Okay. Am I? So I guess just you. Well, and my husband. If you ever want me to do this.
Yeah, I'm willing to meet up during the week too to study stuff or work on stuff too.
Yeah, same here, because I'm here.
You're here in Rexford as well.
Hmm.
Okay. Yeah, I think it'd be nice to have like a study group.
Make it feel a little bit less like an online class. For me.
Yeah, for sure.
So do we individually copy and paste the link or just one of us?
I think it's
Well,
just
another
one
person has
person.
already done it, and it was just one.
One thing we could do though is I'll post it, and I don't know if it would help her as far as grading, but if everybody were to comment on it or reply to it and just say I was in this group.
Okay, yeah.
Okay, good idea.
And so does anyone figure out how to do that one problem?
I just said no, I didn't understand it, or I didn't get it right.
Yeah, I'll put that through.
Will we get docked points for answering honestly?
I don't think so. I think it's just a survey, and I think it's like just to give us like extra feedback and help that we need.
It says you're
Okay.
good for completing it.
Yeah.
So what did everyone get for number 4? I heard the last 2 what everyone got, but what was the first 3?
The
I
first
don't think
first one's the same as the last one.
Okay.
Oh, I just submitted that you do get docked.
Oh, you do?
Yeah.
But that might just be saying what, I don't know if she grades it like that though. It might just be because it's an iLearn quiz.
Oh.
Well, only time can tell.
Let's see.
That's fun.
Well, I think if we put that we get 4 wrong, it's better because then she knows everyone got it wrong. And she,
Yeah, true.
Yeah. And so what was the second and third one for number 4?
so what I put was for the second one, 96 chromosomes, 48 homologous chromosomes, and 24 sisters.
Why is it 24 sister ones?
I don't know. This is what I have.
This is other mitosis analysis.
This is
What?
nothing.
I don't know.
I don't have a good explanation.
Because I understand why it would be 96, and I understand why it would be 48 homologous.
Well, it's because everything got duplicated. So, okay, so are you asking why it's 24 instead of 48?
Yeah.
Okay, See, I don't know that either. I don't know how you count sister chromatids. When I say 24, what I mean is 24 sisters in all 48. I don't know
Oh,
if that's supposed to write it.
how does that work though? So if there's 96 chromosomes total
Yeah.
and 48 homologous pairs, and then within each of those 48, so
Because it
each,
was all doubled.
yeah, but so within the 48 homologous chromosome pairs, shouldn't there be one of each sister, like, you know what I'm saying? Like a sister chromatid for each one, so shouldn't there be 40?
Oh, yeah.
I
Like
know
that's
what you're
what,
saying.
yeah,
I think
like I
you're
just don't understand why it would be 24 instead of 48.
Okay, I agree with you. So I
Like,
think for the next one, in telophase 1, that one should be 24 because we only have 48 chromosomes. But for
yeah.
the beginning of phase 1, we have 96, so it should be 48.
I'm just like thinking in terms of like if you drew it out, if you literally drew like 48 homologous pairs, each of those chromosomes would have like like a sister chromatid. You know what I'm saying? Like there would be a sister chromatid here, and then on the matching one there would be another one. So there should be 48 pairs of those as well. I don't know. I don't know. That's just my guess, but
I think that sounds right.
I don't know though. I could be wrong. I'm just thinking. That's just like my train of thought. I don't know. But I can see for telophase, like, yeah, for telophase 1, why it would be 24. That makes sense to me. But if there's 48 homologous pairs, then I feel like there should be 48 sister chromatids too. I don't know.
So what would the third one be? Would it be the same thing as metaphase, but with just the end of the 24?
Well, telophase is when they
It's when it
split.
splits.
Yeah. So then you would have. Wait, how many diploid chromosomes would you have then? Or how many, I mean, total, what's the total number of chromosomes in it, the end of telophase 1?
48.
It'd be 48 because,
Because they're split,
yeah.
Yeah.
like, completely. Okay.
And then it would be 24 and then 0.
I put 0 homologous and 24 sisters. I don't know. Actually, I'm just not saying the answers, but I think you're right.
If it was the end of telophase 1 and they had just split, I think there would be. Wait, there would be 48 chromosomes. Why is there no homologous pairs, though?
I don't know.
Because doesn't it always have to have homologous pairs?
Mm-hmm.
So I don't know. I don't know. It's a hard one. I don't know the answer to this question either. Sister chromatids. Are those numbers ever 0?
Yeah. Because a sister chromatid is, it's when it's not together, I think.
Oh.
So it can be 0.
Okay. Like when they're split apart, when they're like, yeah,
Yeah.
okay. Okay, that makes sense. I don't know for sure on these ones. I think we'll probably just have to ask her.
Yeah, let's just do that.
Yeah. Other than that, though, I feel like this went well.
Yeah, it's just on the applying genetic problems, 3 and 4 are the same thing as this one, so just in word form.
Hmm. Okay.
Yeah.
Oh boy. Oh.
So any more questions?
Yes.
Did we ever figure out
Did
to post the link? Do we all do, or?
I thought one person posts it and then we comment.
Oh, okay.
Yeah, one person does, and then we commented on it.
Okay.
And it's just under the course introduction group collaboration.
Yeah.
Yeah.
Okay.
And I'll put it up as soon as it's done processing or whatever it does.
Cool.
Okay, I'm going to hang up now.
All right, cool.
Same. This is a good time. Bye.
Thanks, guys.
Bye.